Group number 23. Someone's students from group number 23.
Yes, ma'am. I am present, but I'm not leader.
Not group leader.
Yes, ma'am.
So, okay. The next group number 24. Group number 24.
Present, madam.
Yeah. Present. What is your name? Group leader. Group number
I'm not
24.
leader.
Call me please your name.
Students are joining.
In your group on the lecture can be present 14 students. I hope they will attend. So the next group number 25, 13 students can be present on the lecture. Group number 25.
I'm here,
Madam. Group leader.
Yes.
Very good. How many students now present on the lecture? Do you inform all of them?
Yes, they are joining.
Okay, I will be see. The next group number 26.
Here, madam.
So in your group also 13 students. All are present.
Yes, 6 present right now.
Okay, later I will be check again. Group number 27.
Here, madam, 27.
27, 11 students can be present, yeah? So group number 28. Group number 28, group leader present?
Yes, of course. Yes.
Okay, very good. 12 students can be present on the lecture, yeah? I hope so they attend.
Not all, I think.
Why not all?
I'm asking here in the group. I'm not sure.
Okay. So I hope in a time will be attend. The next group number 29. Group number 29. So repeat, please. Someone's present from group number 29. Okay. Abdullah Nashad, 12 students can be present on the lecture. Check, please. Okay, and inform all of them. So group number 30. Group number 30, 14 students can be present, yeah?
Yes, madam.
Later I will be check. Group number 31.
Yes, madam. Hello.
Yeah, right. So, and don't forget that tomorrow you will have the first class in genetics medical ecology, yeah?
Yes,
so
in
there are 14
biology department.
students at the Department of the Biology, Medical Genetics and Ecology, yeah, of course, with the coding of their official timetable, I hope you know, yeah?
Yes, madam. Yes.
So there are 2 groups will be tomorrow. One of the group will be at 8 o'clock, the another one at 11:30, yeah?
Yes.
Group number 32, present.
Yes, madam.
32, 13 students can be present on the lecture now, yeah?
Yes.
So, okay, group number 33.
Present, madam.
So 12 students can be present. And group number 34, 14 students, yeah?
Yes, madam.
So, and in general can be present 151 students. Now in a Zoom, just a moment, I will be check. Only
Excuse me, madam.
72 students. One half of the students.
I'm from group 25.
Yeah, I'm listening to you. What?
My name is Felipe Dantas.
So now please, all group leaders during the lecture, check please the students in your group and write down in the chat number of your group and the name of the students who's not present in the lecture. Only the name of the students who's not attended the lecture in the Zoom. Understand me?
Yes, madam.
please don't forget about it. If not, will be present the information in a chat about the students who's absent on the lecture. For all students in a group, I will be put absent. Do you hear me? Please don't forget about it. And now again, try to connect with some students who's not linked to the Zoom. So I think that enough to wait, and we need to start the lecture about the genetics of multifactorial disease. And today we will give the characteristics of some aspects of the genetics of cancer or oncogenetics. The main questions. So, excuse me, just a moment. So the plan of our today's lectures are, first of all, we will give the characteristics of disease with a hereditary predisposition. Second question, characteristics of the genetic methods in the diagnosis of multifactorial diseases. After that, we will give the pharmacogenetic approach to the treatment of hereditary pathology. The next question, molecular mechanisms of carcinogenesis, and clinical and genetic characteristics of familial cancer syndrome. So, multifactorial or complex diseases, the one type of form of the general pathology, and which depends from some side, depends from the special genetic composition of an individual. In another side, depends from the influence to the individuals of some risk environmental factors. Many common health problems are due to the interaction of inherited genetic information, which makes an individual more likely or predisposed to developmental disorder if exposed to a particular triggering environmental factor. The environment includes the physical and chemical elements to which we are exposed before birth or after birth. These conditions are called multifactorial, since they are due to a combination of factors. Multifactorial or complex diseases are most numerous and clinically diverse group of human diseases that development with a complex interaction of the genetic and environmental factors. The complex pathology of multifactorial disease, widespread prevalence from the common pathology, make up about 24% of all human somatopathology. The dominant contribution to mortality and disability of the population, as well as a lack of understanding of the causes and mechanisms of the formation of a disease, determine their high medical and social significance. So, designation complex disease is more informative than the traditional designation multifactorial, because the latter term focuses on causative agents, genes, and environment, but not on mechanism or pathogenesis of the disease. The term complex requires that one think about the causative agents and about physiological mechanisms, including evolution, development, and homeostatic processes. In case with the characteristics of the pathogenesis of complex or multifactorial diseases, we will be used the several special genetic terms. The first one, multifactorial. Non-Mendelian character depends on 2, 3, or many genetic loci, with greater or smaller contributions from the environmental factors. Characteristics determination may be due to a single major locus with multifactorial background. Multifactorial in general, determined by multiple genetic and non-genetic factors. Oligogenic, what does it mean? Oligogenic. Characteristic determination involves a small number of genes acting together. A situation in which several genes with moderate effects interact to produce the observed phenotype. And the next, polygenic type or polygenic inheritance. Characteristic determination involves many loci, each individual locus having only a small effect. The term polygenic is generally used to describe multiple factors that are exclusively genetic. So it can also be used in connection with a specific type of model involving multiple genetic factors. Phenotypic traits, which will be described in characteristics of the phenotype of the disease, and any phenotypic traits are determined by the interaction of environmental and genetic factors. There are two types of the traits. First of all, qualitative traits have pronounced forms. There are no intermediate forms. For example, in simple case, it is gender, male or female, or RH factors, positive or negative, either one or the other. Qualitative traits in common have the monogenic nature of inheritance. I hope you understand that in case with a monogenic inheritance, one gene is responsible for their manifestation. And the another type of the traits, quantitative traits. The examples in normal, the traits like a body weight, weight of the brain, mastocytes of internal organs, height, number of blood cells or corpuscles, degree of skin pigmentation, general intelligence, and etc. have quantitative or continuous variability from the minimum value to maximum values in different individuals. There are intermediate options. Polygenic inheritance involves in the determination of the quantitative traits. Does mean many genes are responsible for inheritance. At the same time, the environment also influences the development of quantitative traits. On this picture, you're going to see the polygenic background and disease. When a combination of the effects of the several genes, it involves into the physiological convergence and into their formation of the phenotype of the disease. The genetic basis for polygenic disorder consists of several genes are involved in the ultimate expression of the trait. No single gene shows dominance effect or recessive effects. All of the genes act in an additive manner. Each adds or removes a small amount from the overall phenotype. The genotype and environment interact to produce the final phenotype. In case with the characteristics of the multifactorial traits and multifactorial complex pathology, we will be use their term heritability, the proportion of the causation of a character that is due to genetic cause. Heritability is simply the measure of genetic influence on a trait. If the trait is due purely to genetics and environments plays no role, the heritability is 1 or 100%. If genetic plays no role, then heritability coefficient can be equal for 0. Heritability is expressed as a proportion of 1 or the percentage. For example, in schizophrenia, heritability is about 0.85 or 85%. The normal heritability coefficient for normal trait, for example, for human fingerprints, total ring counts, demonstrate a heritability of 95%. That means that 5% is determined by environment. And if we will be need to give the characteristics of the genetic and environmental factors and its role into dissemination of the thumb pathology, we will be use the calculation of the coefficient of the heritability, and you may to calculate the approximately role of the environmental factors also. So, the concept of liability or susceptibility to disease. The concept of liability indicates a person's genetic and environmental predisposition to developing a disease. If a person's liability exceeds the threshold for a particular trait, then he or she manifests the disorder. Thus, according to this model, all of an individual's genetic and environmental disease predisposed factors, when considered together, constitute liability. On these graphs, you may to see the distribution of their liability to the condition of is polygenic and normal distributed, green curved in normally. People whose liability is above a certain threshold value are affected, for example, here. And their SIPs, it's shown by blue curved, have a high average liability than the population in general, and means a greater proportion of them have liability exceeding their threshold. Therefore, the condition tends to run in families. Characteristics of the liability of threshold model include the next ones. The factors that influence the development of a multifactorial disorder, genetic and environmental, can be considered as a single entity known as liability. Liabilities of all individuals form a continuous variable which has a normal distribution. The curve for relatives is shifted to the right compared to the general population, like the previous picture showed. The closer the relationship to greater the shift to the right threshold exists, above which the abnormal phenotype is expressed. In the general population, the proportion beyond the threshold is the population incidence, and among relatives is the familial incidence. So, in case with the characteristics of the complex multifactorial diseases, we will be use the phenomenon clinical polymorphism. So, it is variation in clinical picture of disease. Variation in the age of manifestation of disease in different individuals. Variation in the person's presence or absence of symptoms or syndromes of the disease in different patients. Variation on severity of disease. Variation in clinical course of disease. And variation in the responses from therapy on disease, individuals' acceptability to management of disease. So we known about their levels of clinical variability in complex pathology. The first of them, inter-individual variability. The next level, intra-family variability, presence, there are changes or the various clinical symptoms in one family, in different members of one family. So we deal population in familial variability when presence there's some changes in manifestation of the pathology or in clinical symptoms for one nosological form, for example, in case with arterial hypertension between the different families, yeah. And the last global level, interpopulation and interethnic variability levels. So, causes of clinical polymorphism based on additive gene interaction, a form of interaction between non-allelic genes situated at different loci when the effects are summarized to produce a phenotype. This phenomenon is a polymery situation when the phenotype is controlled by effects of multiple genes. The next phenomenon which involves into the formation of clinical polymorphism named epistasis. Epistasis, a form of interaction between non-allelic genes situated at different loci, where one gene masks or prevents the expression of another gene at a different locus, or otherwise affects expression in non-additive manner. Contrast with the dominance, the term used when one allele masks another at the same locus. So, causes of clinical polymorphism based also on the genetic heterogeneity, a situation in which distinct mutants alleles leads to the same disease phenotype. Determination of the same disease or phenotype by mutation at different loci. So, another phenomenon which involves into the formation of clinical polymorphism, allelic heterogeneity, also called intralocus heterogeneity, a form of genetic expression in which distinct mutant alleles at the same locus lead to the same disease phenotype. The extent of many different disease-causing alleles at a locus. And another phenomenon named non-allelic heterogeneity or interlocus heterogeneity or locus heterogeneity, a situation in which alleles at different loci leads to the same disease phenotype, determination of the same disease by mutation at different loci. And during our practical classes, we will give the characteristics of the hereditary predisposition, like the presence of the polymorphic allele in different genes, which may involve into their pathogenesis of the same complex pathology. So, incomplete penetrance, what does mean it? Refer to the failure of a genetic trait to be evident even through the genotype that is usually predictive of the phenotype is present. And pleiotropic effects, the occurrence of multiple diverse effects resulting from a single gene, effects on multiple traits due to a single gene. I hope you know about the Marfan syndrome. It's the typical examples of the pleiotropic effects when one gene may detect the formation of the different classes or forms of the symptoms of the pathology. And pleiotropy also involves into the determination of and involves into the formation of clinical polymorphism of complex pathology. So, epigenetic effects, very important ways of the formation of the clinical polymorphism. epigenetic effects, heritable changes in gene expression that is not associated with alterations in the DNA sequences of a gene. For example, a gene may be inactivated by mutilation of its DNA. Gene and genome presence and normal have this normal structure, but not expressed because it depends also from the epigenetic regulation of the gene activity. So, polymorphism, genetic polymorphism, what does it mean? Polymorphism does mean the presence of two or more allelic forms of a gene in a population. At least two alleles with the frequencies greater than 1% must be present in a population. In simple case, we deals with the diallele loci, for example, present the dominant allele of the gene and its recessive allele. For some genes, may be multiple local presence, etc. So, general characteristics of complex disease, we need to call this disease like the disease with their hereditary or genetic predisposition. The base of the disease is gene polymorphism. Hereditary predisposition may have monogenic or polygenic inheritance. Genetic polymorphism or genetic diversity does mean nucleotide variation of genes. 99.9% of genes are similar across people, but the variation involves only in 1,001%. So, there are two main categories of multifactorial disease can be distinguished by their type of the inheritance. Monogenic form and polygenic form. Monogenic form associated with the mutation or with their polymorphic locus only in one gene. So, polygenic form, development with their joint additive interaction of many genes, polygenic inheritance and polygenic form of multifactorial diseases. Genetic heterogeneity or polyetiology, conditions when clinical similar disease in different families and in different patients are determined by different genes. So, more special characteristics of the polygenic variants and monogenic variants of multifactorial diseases includes next one. Polygenic forms are more frequent than monogenic disorder. Approximately 95% of all complex pathology like has the polygenic inheritance. Also, these forms comprise the major contributors to morbidity and mortality in developing countries. Monogenic variants are individually rare, and even in the aggregate constitute a much smaller burden of disease and death than do polygenic variants. So, polygenic variants expression is influenced by the products of multiple genes interaction with environmental factors through the development, maturation, and aging. So, in case there is a difference between the polygenic and monogenic variants, quantitatively in the multiple genes specified products whose effect combine to produce a phenotype in the former, while in the latter the products of one locus override the effects of products of the other loci. monogenic variants complex in their own right, but not as multifarious as a disease that involve multiple genes and multiple environmental variables. Although monogenic and polygenic variants differ quantitatively along a continuum, they don't differ qualitatively. The relationship between genes, proteins, and biological processes is the same in both types of disease. For example, in the next lecture, I will be explaining for you the main differences between the monogenic and polygenic variants of arterial hypertension. The common pathology, arterial hypertension, but the different diagnoses and different pathogenesis may be in different patients. So, polygenic variants require the combination of a number of the unspecific environmental factors. These factors influence all peoples with different combination. Monogenic variants require specific similar single specific factors of usually required for rising diseases and may be expressed regardless of the environment, whether cellular or external. So, in monogenic variants, it often is either too distant to relationship between gene and phenotype. And we know the details of that relationship for many such monogenic disorder. One gene has a pronounced main effect in producing the phenotype in question. Relationship between genes, gene products, and phenotype is less easily distant in polygenic variants. There is no direct relatedness between phenotype and some special given gene. Genetic variations associated with a common complex disease occur in polymorphic genes and are older in evolutionary terms than are those associated with a more rare single gene disorder. Research in molecular biology demonstrates the conservation of important genes, for example, those associated with development and adaptation. So, monogenic variants generally disrupt homeostasis in significant ways early in development. If one gene product is so deficient or dysfunctional as to damage badly the system of which it is a part, that disease must necessarily be rare. It will nearly always have early onset and will resist efforts to provide any specific treatment or even management. Polygenic variants also disrupt homeostasis in significant ways, but the effects are more gradual, often culminating in onset later in life and likely will be more susceptible to treatment. The regions of polygenic variants in any given individual may have a very long history, dating even to intrauterine life. So, monogenic variants arise and manifest before 40 years, borderline age, to discriminate monogenic and polygenic variants due to strong phenotypic effects of single gene mutation. Genetic contribution is up to 90-95%. Monogenic forms have the express specific clinical picture, serious course of disease predisposed to early complications. Polygenic variants usually rise after 40 years due to moderate genetic effects and environment influences during the life. Polygenic variants have the variable clinical manifestation, usually linked to moderate course of disease predisposed to late complications. Familial clustering disease according to Mendelian type of inheritance, usually autosomal dominant or recessive type of inheritance. For example, we will give more detailed characteristics of monogenic form of ischemic heart disease, for example, familial dyslipidemias. More form of familial dyslipidemias have the autosomal dominant type of inheritance, but also may be polygenic some form or maybe have the autosomal recessive type. And of course, we can to know about how we may to distinguish between them. So polygenic variants are inherited by additive polygenic manner. There is no characteristics patterns or pedigrees to demonstrate complex inheritance. There is strong similarity in the clinical manifestation of monogenic variants. Usually all affected relatives have the identical clinical picture of disease. There is, in case with the polygenic variants, there is a significant clinical variation of disease in pedigrees. So, in case with the polygenic variants of complex diseases, often we deal with the principles of anticipation. Does mean consider the tendency for the severity of a condition to increase in successive generation. Anticipation does not characterize monogenic variants. Many polygenic variants tend to be combined with each other due to common polygenic backgrounds of this disease. Such disease combination and so-called as associations. Combination of the some markers, genetic or phenotypic with disease can be also considered as associations. And today later, we will give the characteristics of the some special method for diagnosis of complex pathology, and we will give more detailed characteristics of method of analysis of association. So, monogenic variants usually have no association because condition is attributed to single gene mutations. Positive response from the traditional therapy is an important feature of polygenic variants. There is a resistance to traditional therapy present in monogenic variants. So, the next question of our lecture, genetic methods in the diagnosis of multifactorial diseases. But in case with our form of the lecture, we need to have the 10 minutes for the break, but I think that 5 minutes will be enough because we'll lose the time before the starting of the lecture. And now, please, and during the breaks, I want to remind to the group leaders, don't forget to write in the chat the name of the students who's not present on the lecture. And I see there are 6 group leaders wrote it. Do you hear me, guys? Abdullah Nashat, do you hear me? And other all students, I hope you. Okay, 5 minutes break, and after that we continue. So the next question of the lecture, genetic methods in the diagnosis of multifactorial diseases. Do you hear me, guys?
Yes, ma'am.
Okay. So now present 83 students only on the lecture. 83 students. Mhm. So the first method in molecular biology and in medical genetics, which was used for detection of the primary causes of the disease, was southern blot hybridization. Now not used today, but if some students will be interesting, we need to have some reporting during the class about the oldest methods in medical molecular genetics, southern blot hybridization. It nowadays more actual method, polymerase chain reaction, a method widely used to rapidly makes millions to billions of copies of a specific DNA sample. PCR, or polymerase chain reaction, is now a common, often indispensable technique used in medical laboratory research for a broad variety of applications, including medical research and criminal forensics. Another method is reused enzymes such as endonuclease that recognize specific DNA motif and cut them. The method named restriction fragment length polymerase analysis, or RFLP analysis. And after that, we made to detect it by electrophoresis in the some genes, and if present the some DNA motif and it present the cutter in this place, will be shown like there are two bands. Not present does mean only one bands. So on this picture present there some natural photo of identify two point mutations or polymorphic locals in a real and mean and acetyltransferase gene. The one sample, second sample, third, and sample does mean the sample it is there of one individual. If present there restriction, present there are two bands. If not present the restriction or not present the variation in a gene, present only one band, maybe homozygous C in this case, or homozygous T genotypes only present in some individual. So the next direction in their medical genetics for the determination of the hereditary predisposition to the complex disease, DNA microarrays using the DNA chip. Science use the DNA microarrays to measure the expression levels of large numbers of genes or to genotype multiple regions of a genome. An example of its application is an SNP arrays for polymorphism in cardiovascular diseases, cancer, pathogens, and GWAS analysis. A method genetic analysis that compares the frequency of alleles between affected and unaffected individuals. We talked about the association analysis, and the molecular genetic methods are being used for studying of multifactorial diseases in several studies decisions. First of all, association analysis. And a given allele is considered to be associated with the disease if that allele occur at a significantly high frequency among affected individuals, like at present in this diagram. In comparison, the frequency of markers in the samples of patient and in samples healthy controls or like a general population, often using some special candidate genes. For example, of the association analysis may be GWAS or genome-wide association study is an approach that involves rapidly scanning markers across the complete sets of DNA or genomes of many peoples to find genetic variants associated with a particular disease. Once new genetic associations are identified, researchers can use the information to develop better strategies to detect, treat, and prevent the disease. Such studies are particularly useful in finding genetic variations that contribute to common complex pathology such as bronchial asthma, cancer, diabetes, heart disease, mental illnesses, and etc. And for the characteristics of the hereditary predisposition, we will be use the information from the Internet GWAS catalog for characteristics of the hereditary predisposition to some special complex pathology. The another way of the methods, it is linkage studies. Linkage. The tendency of genes on some chromosome to segregate together. Linkage analysis, a statistical method for detecting linkage between different loci using families. And linkage disequilibrium, a situation in which the frequency of a certain haplotype in a population is not equal to the product of its respective alleles frequencies. The association between alleles at different loci in the population. On this picture, you may just see there are two different families, the called segregation of markers in some chromosomes, and detected by the special symbols which are present here, and the combination of the some special genes and its form in the some chromosomes named the haplotype. So, some are epidemiological, and nowadays, for example, Director of National Office of Public Health Genomics, Moon Knuri, told that genetic and epidemiological research sets itself the task of assessing the role of genetic factors and their interaction with environmental factors in the occurrence of disease in human population. And in nowadays, very important studies the environmental risk factors which may cause the formation of the pathology, which may influence into the genotype of some individuals, and may involve into the pathogenesis of some complex diseases. If we will be known, if we will be detected, we need to prevent the formation of some disease. So, their genetic prognosis for multifactorial disease depends on their next factors. The low frequency of the disease in the population, the higher the risk for relatives of prevent. The stronger the severity of the disease in the proband, the greater the risk of developing the disease in his relatives. And the risk to relatives of the proband depends on the degree of relationship with the affected family member. The risk to relatives will be higher if the proband belongs to the less affected sex. So, on this picture, on this table, you need to see the just a moment. There are main direction to expected for to diagnosis of the hereditary diseases, like a type of the diagnosis and analysis of the specific phenotype, which we need to divide it, which detect by single gene etiology, you need to see it here, or multiple genetic etiologies, or broad phenotype. In case with a chromosomal disorder, we talked about it on previous lecture. We deals with the cytogenetics examination, and the first of them needing to study the locus specific, and after the genome-wide association model may use. And in case with a chromosomal disorder, we need to use the karyotyping. In case with another one, we need to use some special methods which are present for demonstration of there are a lot of many different technologies which now we need to use for detection of the hereditary diseases. So, we talked about the genetic prognosis and genetic risk. In multifactorial diseases, the basis for the risk assessment is empirical data on population and family frequency of each of them. Specific genetic risk, less than 2 to 5 persons, is considered to be low. 5 to 10 persons increase to a mild degree, up to 20 persons medium, above 20 persons, genetic risk will be high. Genetic risk of moderate to high degree is regarded as a contraindication to conception or indication to terminate an existing pregnancy. The examples of the genetic risk for the some relatives risk in case with the familial breast cancer, you need to see the approximately population risk, 1 among 12, cancer in a sister under the age of 65, 70 years, 1 among 8, and etc. On the next slide, you need to see there are some genetic markers which we need to use for diagnosis, prognosis, or prediction of multifactorial disease. Genetic markers does mean there are some special genes, candidate genes of the some pathology. For example, we know that the main genes in our case with the type 1 diabetes mellitus, it is a system of HLA system genes which determine, and we need to detect in the some patient the polymorphic loci in this genes for the prediction of the formation of the diabetes mellitus and etc. The next question, which directly depends from the knowledge in molecular genetics and based on the knowledge of the functional and structural organization of the human genome, pharmacogenetic approach to the treatment of hereditary diseases. Pharmacogenomics is a study of the role of the genome in drug response. So it deals with the influence of acquired and inherited genetic variation on drug response in patient by correlating gene expression or single nucleotide polymorphism with pharmacokinetics, drug absorption, distribution, metabolism, and elimination of the drugs, and pharmacodynamics, effects mediated through the drug's biological targets. So, genetic characteristics of patients associated with the changes in the pharmacological response are relevant during pharmacogenetic testing. Pharmacogenetic test is the identification of specific genotypes associated with the change in the pharmacological response. These tests are based on polymerase chain reaction, or PCR. The use of modern international information resource on genetics makes it possible to gain knowledge about the role of genetic factors in the shaping the response of the human body to drugs. Efficiency, not efficiency, the development of adverse side reactions, learn to interpret the results of pharmacogenetic testing, predict the pharmacological response to drugs, and formulated recommendation for the choice of drugs and their dose requirement, thereby personalize the patient's treatment. So on this slide, you need to see the several genetic database and their main use of them. PharmVar, supersede bioinformatics tools, PharmGKB, GBSNP database, and etc. It is free scientific database which we need to use during your works, of course. So the next question of our lecture, genetics of cancer. Genetics of cancer. Molecular mechanism of carcinogenesis. There are very few cancers with non-Mendelian inheritance, but which have large seed-relative risks. Common cancer, such as those of the colon, breast, or bronchus, are usually late onset, and the parents of patients are often dead. Many cancers are fatal, making samples difficult to obtain retrospectively and even contemporaneously. Not only does cancer require a particular combination of genes and environment, but it also has a truly random component in the several somatic mutation must occur for a carcinoma or sarcoma to develop. The challenge of discovering genes mutated somatically in cancer, but with non-germline effects, has been a worthy distraction for the genetics from the task of studying cancer as a complex genetic disease. Molecular mechanism of carcinogenesis based on two-hit hypothesis of carcinogenesis, which start from the mutation in germ cells, germ-lung mutation like first events, which increasing the sensitivity of somatic cells to environmental carcinogenesis influences, and acquisition of somatic mutation, second event, and into the molecular mechanism of carcinogenesis involves the oncogenes and proto-oncogenes. An oncogene is a gene that has the potential to cause cancer. In tumor cells, these genes are often mutated or expressed at high levels. Most oncogenes become as proto-oncogenes. Normal genes involved in cells growth and proliferation of inhibition of apoptosis. Now we may give the characteristics of there are four molecular ways of activation proto-oncogenes. First of them, activation by amplifications. Second one, activation by point mutations. The next one, activation by translocation that protest a novel chimeric gene, and activation by translocation intertranscriptionally active chromatin region. So there are two main parts of the genes involved into the formation of the cancer. First of them, genes regulating cell cycle and genes of common controls. The first type genes regulating cell cycle. Germline mutations affect on the genes regulating cell cycle, like we told about the first mutational event. For development of cancer, the second mutation in normal allele should arise, like point mutation, loss of inactivation of normal allele. So genes of common controls, genes support to genome instability by affecting DNA reparative processing, multi-stage process. One dose of mutation does not lead to a cell transformation. Inactivation of second allele of the SAM gene leads to an instability of genes of the cells, thereby predisposing to the carcinogenesis. Subsequent mutations, including within gene regulating cell cycle, accumulated and by that involve malignancy. So, mechanism of the genetic induction of carcinogenesis, the first of them, tumor suppressors. More 20 gene suppressors have been described in literature. Some of them you may see on this table. These genes do control the mitogenetic capacity of cells, where their proteins products which support both normal growth and differentiation of cells. And some polymorphic loci of these genes involves into the formation of some types of their cancer. Second group, proto-oncogenes. About 100 proto-oncogenes have been described. These genes are normal genes controlling their proliferation rate and intracellular signal transduction. Their mutations lead to transformation of proto-oncogenes into oncogenes, which dysregulated the processes of cell divisions and differentiation. The next third group, abnormal DNA reparation. Genes involved in reparation of DNA defects may be responsible for genome instability and carcinogenesis. The products of these genes recognize and repair DNA damage before division of the cells, like the genes which involves into the DNA mismatch repair system in generally. The first one, involvement of chemical detoxifying system in carcinogenesis. Xenobiotic metabolized enzymes play a crucial role in the metabolism of carcinogens. Mutant alleles in xenobiotic metabolizing enzymes increase sensitivity of tissues to environmental carcinogenesis. I want to remind for you the main phases of detoxification of the xenobiotics. Phase 1, metabolic activation of the xenobiotics. Phase 2, detoxification of active carcinogenic intermediates. And phase 2, excretion of inactivated substances from organism. The mutation in the genes which detect the enzymes of each phases involves into the formation in generally in the human cells, into the formation of the oxidative stress, and of course involves into the carcinogenesis. So the next characteristics, etiological classification of tumors. First one, inherited variants of tumors in which single gene mutation is responsible for disease. Second group, multifactorial variants of tumors resulting due combined effects of genetic and environmental factors. Third one, tumors arising due to spontaneous mutation in somatic cells under usual physiological condition without additional effects of environment. And fourth one, tumors arising due to actions of environmental mutagens and cancerogens, which are chemical, physical, and biological. So let's consider the characteristics of the some types of tumors. Nephroblastoma is a cancer of kidneys that typically occur in children, rarely in adults. It's named after Max Williams, and on the photo you might see Wilms tumors. Prevalence 1 in 8 to 10,000. Manifestation onset or 10 to 12 years. Etiology involves inactivation of suppressor gene WT1, Wilms tumor proteins genes. And the second gene which involves into the pathogenesis of nephroblastoma, it is MER1 gene. APC membrane recruitment protein 1 gene. The gene has the X-linked dominant type of inheritance. So 40% patient has germline mutations without autosomal dominant type of inheritance. 60% patient with the nephroblastoma has a somatic cell mutation, which forms de novo, which are not inherited. Criterions of inherited nephroblastoma: bilateral affection of kidneys if present, polyfocal affection in kidneys, and at least one affected by nephroblastoma relative in the family. The examples of the patient with the Gantt nephroblastoma, patient in 5 years, boy. So also you might see in rentgenogram nephroblastoma and computer tomogram, large nephroblastoma at the left side. The next, retinoblastoma, malignant tumor of the retina which develops mainly in childhood from the tissues of embryonic region. You might see the peak of the disease occur at 2 years. Almost all causes of the disease are detected before 5 years of age. Prevalence 1 to 10-15,000 depends from the population. Onset usually during first year of life. Etiological causes: germline mutation, deletion in 12 chromosomes in which are present the genes RB1 gene, or maybe somatic cell mutation and inactivation of the second allele of the RB1 gene in retina. It has the autosomal dominant type of inheritance with 90% of penetrance. The next, neuroblastoma, a malignant tumor of the sympathetic nervous system. Neuroblastoma occur only in children. It is the most common malignant neoplasm in early childhood, 14%. In some cases, it found at birth may be accompanied by birth defects. prevalence 1 to 8 to 10,000. Great majority of cases are sporadic and non-familial. About 2% of cases run in families and have been linked to specific gene mutations. Familial nephroblastoma in some cases is caused by a rare germline mutation in the anaplastic lymphoma kinase gene, ALK gene. Germline mutation in FOX2B or KIF1B gene have been implicated in familial neuroblastoma as well. Inactivation and also maybe involves the inactivation of the suppressor gene, which localizes on the first chromosome. Neuroblastoma has a number of properties that are absent in other malignant neoplasm. Ability for spontaneous regression, the ability to maturation, and ability to rapidly aggressive and develop metastasis. The median age for death is 2 years. Large nephroblastoma on a computer tomogram on photo you may see. The next form, melanoma. Malignant tumor that develops from melanocytes, pigment cells that produce melanins. 3% of malignant tumors among children. Unfortunately, nowadays, etiology is not completely known. In some cases, melanoma occurs as a result of a mutation in the tumor suppressor genes, which encodes some proteins, and the second genes are type 4 cyclin dependent kinase. We know that the mutation in these genes involves into the formation of the melanoma. You might see the examples of melanoma on the skin, on the face, and multiple melanomas on the left leg. So next question about the characteristics of the personal and family history features suggest a hereditary cancer syndrome. So, and characteristics in generally includes age of onset of cancer is younger than typically seen for the cancer, usually before 40 years in adults. Cancer occurring in the sex not typically affected, for example, breast cancer in a man. Biliary cancers in parotid organs development. Multiple tumors in the same organs or multifocal tumors. Synchronous or metachronous primary tumors in the same individual. May also constellation of cancers in the same patients or in multiple members of the same family. Uncommon or rare type of cancer, if we detect, we may to think about the presence of the familial type of the cancer. So, cancers spanning multiple generations that are suggestive of a common cancer syndrome. So, evaluation for hereditary cancer syndrome. The diagnosis of a hereditary cancer syndrome is highly depends on the accuracy of the personal and family history data. Several key points of information need to be collected from the patient to determine the familial nature of the cancer. Tumor characteristics. This includes the type or pathology of the cancer, special characteristics of the tumor, immunohistochemicals, staining, and other ones. So very important to analyze family history. Detailed 3 to 4 generation pedigree should be obtained from the patient. Remember that the risk is independent of each side of the family. So maternal, paternal in this case, and maternal lines. The persons in the patient family of 3 or more cases of various form of cancer among first-degree relatives is considered as family cancer syndrome, which shows on this pedigree. In this family present the ovarian type cancer, colon cancer, endometrial cancer, and etc. Combinations and different types of the cancer. So, scoring tools. Many online software tools are available to determine the risk of finding genetic variants, such as BRCA Pro, PREM, and EBIS. They are often based on the personal and familial history information. Some of these tools may require additional licenses. Also, some can be time-consuming and challenging to implement into busy clinical practice. The next, evaluation diagnostic testing. Once a pretest clinical assessment for hereditary cancer syndromes have been conducted, genetic testing can be ordered. This can include a single gene or multiple genes. Current standards of care for evaluation of hereditary cancer syndromes is testing multi-genes panels. Unless single-site testing is pursued for non-familial variants. So, molecular genetic method which we may and need to use for diagnosing cancer. There are several areas in clinical oncology where markers isolated using genomic technologies are already actively used. So, diagnostics of hereditary form of cancer, determination of forecast markers, determination of markers indicating the initial stage of the tumor process, determination of markers indicating the presence of micrometastasis, and diagnosis of DNA markers that determine sensitivity to the drugs, especially target therapy. On this slide, you may see the list of the select cancer with their associated genes and cancer syndromes. All organs may affected by cancer, by tumors. You might see presence here, the associated genes and the name of the special hereditary cancer syndrome. So adrenal, basal cells, bladder, bone, breast, cervix, colon, endometrium, esophageal, gastric, and etc. One of the most common forms of familial cancer is familial breast cancer. The genetic nature of breast cancer is being actively studied at the present time. Found genes of a high degree of penetrance, aberrant Mendelian laws of inheritance, in an autosomal dominant mode of inheritance, BRCA1 gene and BRCA2 gene. Multicenal clinical trials to identify the risk of breast cancer and ovarian cancer are taking place in different countries of the world. Screening for BRCA1 or 2 genes is recommended as an important marker for early detection of all patients at risk with breast cancer and a family history of the disease. So, familial breast cancer, you might see is the general population risk because presence of the some mutation or negative variants in BRCA1 gene, and risk because the presence the negative variants of BRCA2 genes. So, for example, the family breeds in general population risk approximately 12 persons. If we detect in some woman's mutation in BRCA1, the risk increase to the 87 persons and etc. So, non-hereditary syndromes characterized by the development of the tumors. Tumors often associated with the hereditary syndromes. Multiple endocrine neoplasia type 1, multiple endocrine neoplasia, von Hippel-Lindau syndrome, so very important early prevention of hereditary forms of cancer. We need to make recommendations regarding lifestyle in case of hereditary syndromes, periodic examination of the patient. In some cases, it is advisable to resort to prophylactic removal of a part of the organs affected by tumor, for example, part of the intestine with hereditary predisposition, or breast removes, etc. Genetic counseling in family planning and prenatal diagnosis of onco syndromes. So that's all for our today's topic. Thank you for your attention. Now, if someone have a question, I'm listening to you. I hope you wrote in a chat the name of the students who's not attend to the lecture, yeah? Guys, do you hear me? Aminat Shifra, do you hear me? Aminat Shifra, I put absent for you. Aminat Shifra not listen. Yeah? Ankita Singh. Do you hear me? Ankita Singh. Also absent. So, Vanpati Gopal.
Yes, ma'am.
Okay, good.
Okay.
So please inform all students in your groups, in your course, that I will be check there all students in a group on the next lecture. Today is present only there are 83
Madam?
students on the lecture. Yeah. I'm listening to you.
Yes. Oh,
Amita Shifra, I called your name earlier. Do you sleep now? Or do you have a problem with the connection? I think, yeah. So, if you don't want to ask me about the some problems in multifactorial diseases, thank you again. And now you may be free, you may to switch off from the